Biological evolution could be described into two concepts, microevolution and macroevolution. Microevolution is an evolution of adaptation that can be observed in nature and laboratory. On the other hand, macroevolution is an evolution involving speciation which cannot be witnessed because it only occurs over the time intervals that far exceed the human lifespan. Speciation is a type of macroevolutionary process that also occurs through the formation of barriers to gene exchange between groups of organisms leading to the genetic differentiation of the groups. These barriers are classified as pre- and post-zygotic barriers. Pre-zygotic barriers include all the factors related to mate location, courtship, and behavioral and morphological mating. Post-zygotic barriers include hybrid unviability. Speciation is defined as the eternal disassociation of population systems so that migrants from one system would be hindered when entering another system. The hindrance of entering could be rooted from a lacking of mating partners from a particular population or the failure to withstand to ecological and biological competition and challenges in the environment, thus making the whole concept of speciation a result from either adaptive changes or from changes that are a result of that changes. The origination of species has been the greatest idea of Charles Darwin that has taken evolutionary biologists until now to realize he was actually correct. He looked species more like an individual closely resembling each other. Speciation by natural selection occurs by two general mechanisms. First is ecological speciation, that is defined as the evolution of populations or subsets of population through reproductive isolation leading to ecologically based divergent natural selection. In this process, natural selection's role is contrasting directions between environments, which drives the fixation of different traits. Each is beneficial in one environment but not in the other. The other one is mutation or speciation, characterized as the divergence of populations as they accumulate a different series of mutation under similar selection constraints. In both speciation mechanisms, allele fixation is possible because of natural selection, but divergence is only favorable in ecological speciation and thus only occur by chance in mutation order process. Sexual selection has a reputation of being the major driver of speciation, and that brought surge in popularity because of supporting evidences that tends to be accepted, rather alternative explanations that are not so thoroughly considered. The involvement of success in mating and successful models of sexual selection-driven speciation has seemed to be the reason of the reputation of sexual selection. Sexual selection also predicted by to drive the coevolution of mating signals or mating traits between sexes within populations and could play a role in speciation if sexual isolation happens because of mating trait divergence between populations, but few studies also said that differences in mating traits result from sexual selection alone and not from other factors. Undeniably, sexual selection drives speciation and coevolution, but that is not always the case because as it often shifts traits away to some organisms from their survival optima, it also increases potential population extinction when there are environmental changes, as seen in the experiment conducted in Baker's yeast. The term speciation is also defined as the continuous process of the creation of one or more new species that comes from an existing species. It is said that there are two general pathways that leads to speciation: transformation and divergence. Transformation is characterized as the result of the changes over the long period of time, thus making a whole completely new species, while on the other hand, divergence is when one or two species arise from the parent species and coexisting with each other. These two pathways are a product of natural selection. Divergence fosters biological divergence because of the increase in number of species, while transformation is not, because as new species arise from the old one it also means the loss of it. Others define speciation involving the reproductive isolation changes over time. It is a continuous process and complex, and there is an evolution of multiple interacting reproductive barriers among populations that does not affect the variation across a whole genome. There are several types of speciation that happens over the course of time. According to National Geographic, there are 5 types of speciation, namely allopatric, sympatric, peripatric, and artificial speciation. Although artificial speciation is the only type of it that does not happen because of natural selection but rather by manually achieved in laboratory experiments. The other different types of speciation are characterized by the type of barriers present in the process of the modes of cladogenesis or divergence that occurs during the course of speciation. Scientists simply discuss that these types of speciation is explained by how genetic flow within the population of organisms are disrupted. Allopatric speciation. Russell, Hertz, Macmillan in 2017 discussed that geography plays a huge impact on whether gene pools have the chance to mix. They defined allopatric speciation as a process of which it occurs between geographically separated populations. The word comes from the Greek terms allos and patria, which means different and homeland, respectively. It is the most common type of speciation among large animals. This could take place when a physical barrier subdivides a large population or when a smaller population becomes separated from a species' original geographical distribution. Allopatric speciation occurs in two stages. First, two populations must be geographically isolated and separated to prevent gene flow between them. Second, the population experience particular mutations, and different patterns of natural selection, genetic drift, and there will be accumulation of genetic differences that separates and isolate them reproductively. Allopatric model of speciation is described as a large population that is at first distributed over a large area becomes separated because of any geographical changes brought about by natural occurrence such as tectonic movements, hurricanes and other agents. In this event that the populations are divided, through the course of separation, they evolve independently and diverge into new species with the absence of gene flow, genetic drift and also natural selection. When another geographical event will occur leading to the new distinct species to come to secondary contact. Even if this contact and interaction take place, they will not interbreed. Secondary contact is a period post of geographic isolation and provides a test of whether their gene really diverged enough to make them reproductively isolated. When their genes did not undergo intensive variation during the course of isolation but carrying distinct characteristics, the possibility of species fusion may still occur. In the paper written by Zink, 2012, there are some characteristics needed in the populations to be considered distinct species under three different species concept, and those are geographic isolation, diagnostics characters such as phenotypic, ecological, molecular, physiological or behavioral, and reproductive isolation. He said that all concept of species, biological, phylogenetic and evolutionary concepts undergo allopatric speciation. Both biological and phylogenetic concepts of species develop distinctive diagnostic characterization. Only biological concept of species has reproductive isolation. Sympatric speciation. Sympatric speciation comes from the Greek word sym, which means together. This only happen when members of the same species that are within the same range diverge in two or more different species even though there are no physical and geographical barrier and interbreeding. It is believed uncommon kind of speciation compared to allopatric speciation, especially in animals, but evolutionary biologists found several ways in which it can happen and that include polyploidy and adaptation to local environment. Polyploidy is a type of genetic change that results to immediate reproductive isolation in which organisms has more than two sets of chromosomes. This kind of genetic change is more favorable to plants and not on animals. It has advantages for plants, especially for production. This is a mechanism wherein there is non-disjunction of chromosomes and which increases the number of chromosome sets in the given species. Such changes lead to abrupt speciation. For example, non-disjunction resulting a tetraploid plant was due from the union of two diploid plantar, and when these two are crossed they will result to two sets of triploid plant organism. Triploid organisms are usually sterile in the animal perspective. This hybrid sterility provides reproductive isolation between triploid and diploid organism. Another mechanism is allodiploidy, in which two organisms of very different species but with sometimes close evolutionary relation interbreed and the resulting organism is automatically reproductively isolated from that of the related diploid species. Sympatric speciation in adaptation to local environment is characterized when populations occupying different local locations that are continuous with each other forming different species. Example for this one is when biologists from America studied North American apple maggot fly. This fly originally fed on hawthorn trees, however when apple trees are introduced in the area in nearly 200 years ago, this provided new environment to the species. Apple-feeding fly develop more rapidly because apple trees mature faster than hawthorn trees. This results to partial temporal isolation. Scientists said that although they are subspecies, they will eventually become distinct species due to reproductive isolation and independent mutation. In the argument provided by Bolnick in 2011 to understanding why sympatric speciation is rare or absent in some organism, he provided some simulation results comparison from 3 spine stickleback. He said that sympatric speciation occurred in at least few cases but still relatively rare and difficult to prove but theoretically plausible and needs specific conditions. Comparison between the experiment on theory and empirical parameters were taken to prove the existence of the speciation. Furthermore, existing models made various related testable predictions and observations such as the links to competition, habitat diversity, and sexual dimorphism. Core assumption in sympatrism is qualitatively reasonable but speciation in this sense may have failed because of quantitative mismatches with theory and alternative evolutionary outcomes. Parapatric speciation. Parapatric speciation is the process of species formation in the presence of some gene flow between diverging population. It represents the most general scenario of speciation, which includes both allopatric and sympatric speciation as extreme cases. The geographical structures of species that has many local populations experiencing genetic contact for a long period of time implies the parapatric speciation event. Although this may be real and plausible, this type of speciation still skeptically receiving little attention compared to a large number of theoretical and empirical findings all due for allopatrism and sympatrism. Parapatric speciation come from the words para meaning near and patriato meaning country and only occur when a smaller group is isolated from a larger group and becomes differentiated to the point of becoming a whole new species. The possibility of gene flow remains in the two populations during the process of speciation, and hybrid zones may be observed at the interference between the populations as a result. Example of this is the population of the grass Agrostis tenuis, which thrives in mine tailings and normal soils. There are species that are tolerant in heavy metals and contaminated soils, and reverse happen to the which are intolerant to the mentioned soils and only grow to the normal soils. There is gene flow between subpopulations on and off on the mine tailings, and hybridization happened because of their slight difference in flowering time in those two locations. Since in parapatric speciation is possible, there is this limitation found by Blockhart and Hermison in 2018. They analyzed the genetic barrier by genetic incompatibilities to gene flow could evolve when there is migration. They found out that new mutation brought about by migration actually destroy that barrier, and effect is disrupting the snowball effect that leads to allopatric speciation. Parapatric speciation. Rutledge and others in 2011 described parapatric speciation as a process of forming new species resulting to smaller groups of individuals that break off from the larger group, just like in allopatric speciation, and physical barrier makes it impossible for interbreeding. Certain and unique characteristics of the smaller groups are passed to future generation of the group, making it more common to them and distinguishable from the others. Parapatric speciation is also evident on the findings done by Valchuina and others in 2017 on an endemic Macaronesian plant after recent divergence from a widespread relative. This group of scientists conducted study between Scrophularia loi and Scrophularia arguta. They said that S. loi arisen from S. arguta. They had 5 species of S. loi and 25 species for S. arguta and analyzed the possible differences in their morphological, nuclear DNA, and chloroplast DNA. Results showed that these two related species differed in morphology and nuclear DNA but not on their chloroplasts, supporting their claim for baropatric speciation. They said that the new dispersal of S. arguta to Madeira from Canary Islands or Salvagens Islands and the swift morphological mutation and differentiation after the colonization to generate the S. loci is the plausible explanation of the emergence of the last taxon. In animals, heripatry is also observed by researchers Lawson, Bates, Manigan, and Loader in 2015. They said that understanding the role of different speciation mechanism is vital in understanding the cause of biodiversity. In their study, they used spiny-throated reed frags that undergone phylogeographic analysis conducted by them. The baropatry was observed through the uneven potential and realized distributions and uneven population size estimates on the field collections and climatic shift that causes the split of the frog species. Sexual dimorphic traits such as their body size and gular shape was also considered as the differentiation of these organisms. They inferred that baropatric speciation was the significant driver of diversity of these species in a fragmented ecosystem. Heripatриc speciation evidences were also concluded to the diversification of some insects. Tseng and others in 2017 conducted a study on flightless Pacrentius orbifera, a type of weevils. They investigated the spatial and temporal patterns of diversification and colonization of these insects across Taiwan-Luzon volcanic belt. They said that the probable factors shaping the colonization history of the P. orbifera was due to the nonlinear, more complex spatial and temporal setting of the archipelago and that speciation occurred only between islands, and baropatric speciation through the founders of stochastic dispersal was the major evolutionary front-runner.